See, can you read this name here?
No, can't. Well, you see. No, that's okay, Rajan.
Oh, they have a presentation.
Huh? What is that, Rajan?
Someone is showing you something.
So I have to click?
No, just wait.
But he says click to extract.
No. See, that's on his screen. Don't worry about it.
Oh, we are back.
Now I want to know, sir, may I know who you are?
Who?
This one.
Oh, I don't know. His name isn't there.
No,
Hello, the name is unknown. May I know?
I'm waiting for a
Must be me, Krishna Kumar. My name is Krishna Kumar.
Yes.
You are addressing me. I am Krishna Kumar, and I was there in 84 to 86.
So you are the one who said you were there in 1984 to 86?
Yeah, that's right.
That means you were just ready to kick me out. I was there in 1986. I left.
Yeah. I was there from
And
84 to 86.
of course, Sarita, I can see her laughing. And then John Zakaria.
Yes, sir.
You know,
1978, 80, sir.
78 to
80.
Are you in ISER Pune?
No, sir. No, IAS, sir. Calicut, Kerala.
Oh, now you are in Calicut. Oh, no. I know Zakaria. Yes. You are from Calicut, right? I remember. But then you have become much older than me, so I'm not able to identify you. I'm sorry. No, I remember Zakaria, and of course, Ratna Prabha. You see, isn't it? Am I correct?
And Tata, there are 15 other people. There are 15 other people we can't see.
I see. So we can't do anything.
Yeah.
That doesn't matter.
But I think maybe when they talk, they will appear.
That's fine. And, so this is Pushpa. This lady I know very much because she was always with Sarita Gupta. Used to entertain me whenever I went and then shower all the gift on me. So I know this lady very well, you see. And of course, our Arco, you see, the big man now. How is your health now? Is it all right?
All right now.
That is more important for me, you see. So, and this is myself. You see, I was wondering there is somebody else here. Then I found it is myself, you see.
Yes.
So, sir, now I am just ready to hear whatever you people are talking.
Yes, CVR, sir.
I think you are going to start his lecture now.
Yes, sir.
So good. You can do that. I can
He
hear.
has just joined. We'll start with the university song.
Yes. Yeah, you
Yes,
can do everything. I will just observe. Yes.
Tata, I will go now. If you need me, just call me. I'll come right away.
But you are going to be in the house.
Yes.
Very good. Okay.
Daniel,
You
please start the
see,
university
but
song.
for my grandson, I cannot operate the computer. I know nothing. You see, so, okay.
Hey, he's pretty impressive. He's 96 and he uses Linux.
विद्यापीठना सपना सारवनारा हमें ज्योति जलावी सृष्टिनावली सहसा सर्जनहारा हमें बड़ोदराना विद्यापीठना सपना सारवनारा हमें ज्योति जलावी सृष्टिनावली सहसा सर्जनहारा हमें गगन कुसुम कर धरनारा हमें मगन थी फरनारा मगन बाज हमें धरनारा हमें दैन्य मिलने हरनारा हमें बड़ोदराना वेद विद्यापीठना सपना सारवणारा हमें ज्योति जलावी सृष्टि नगरी सहसा सर्जनहारा श्री सयाजी विद्यापीठना ज्ञान दीपने धरनारा श्री सयाजी विद्यापीठना ज्ञान दीपने धरनारा सत्यं शिवं सुन्दरं मन्दनन्दन हरनारा
Thank you so much for that. We will begin our program with the welcome address by the Dean, Faculty of Science. We acknowledge with gratitude the presence of our Dean, Professor Sarabhai Kataria, a young dynamic mathematician who has infused the science faculty each year. Professor Kataria joined as a Dean in 2018. He is a professor of mathematics and his research interest lies in the mathematics of fluid flow mechanics. He is an astute administrator and has served the university in various positions. As Dean of Science Faculty, he leaves no stone unturned to improve the academic standards of our faculty. He has managed to continue the teaching semesters without break during the pandemic. He ensures ease of work of the faculty and is always approachable to the students and staff alike. We are very thankful to you, sir, for being with us today, and we invite you to give the welcome address.
Thank you, Professor Pushpa, madam. Am I audible?
Yes, sir.
Yes, sir.
CVR sir, good morning, sir. Am I audible, CVR sir? Good morning, sir ji.
Good morning, sir.
Sir, happy to see you. It's a matter of pleasure for me, sir. Good evening, everyone. It is indeed my privilege to welcome one and all for this unique evening. We all know that we are gathered here virtually to celebrate the birthday of one of science faculty's oldest professor. Sirji, many happy returns of the day. I had the fortune of interacting with Professor CVR during his last visit to our campus.
2019.
But the moment we met in the cell and molecular biology department, of course, some of your proposals I couldn't through it because of one or the other reasons. But I am behind these proposals you have suggested, sir. See, he always takes keen interest for the growth of our university and in particular the growth of the Department of Biochemistry and CM. Because both are his children, biochemistry as well as CM. Has helped us with the CMB program with his vision and farsightedness. Thank you, sir, for this virtual gathering and wish you good health and days to come. I take this opportunity to welcome Professor R.K. Rao seminar and look forward to meeting him in person when the pandemic is over. Professor R.K. Rao, we welcome you, sir. I welcome all alumni of Biochemistry and CMB for this virtual seminar and urge you to stay connected to your alma mater. We look forward to mutually enriching interactions with alumni. I invite you all to come forward and reach out us. wishing you all the very best in this new year. I take this opportunity to thank the organizers for inviting me and organizing such a wonderful evening for our beloved Professor CVR sir. Thank you one and all present. Thank you so much.
Thank you, sir. So today's event is mainly possible because of the Alumni Association, and the Alumni Association of Biochemistry Department is mainly because of Professor Harish Pad. Professor Harish Pad as Vice President, Professor Tara Mehta as President, have done a lot to keep the biochemistry family together. I request Professor Harish Pad to say a few words on this occasion.
Well, fellow colleagues, alumni of the Department of Biochemistry, MS University, I won't take much time. I just want to greet Professor Ramakrishnan on his birthday today. On behalf of our President, Professor Tara Mehta, on my behalf and on behalf of all the alumni of the department, sir, we wish you well. We wish you a good health and longevity much beyond the century. And we look forward to see you celebration of your 100 years in the department in Baroda. And we really wish you that you will have a very healthy longevity much beyond 100 years. Alumni Association it was started somewhere in 1997-98, primarily because of the motivation that Professor L.J. Parekh provided. And he brought out his various diaries, and from that diary, we compiled the names and the addresses. At that time, there was no internet. So we collected name and addresses from his diary primarily. And we created Alumni Association way back. And the prime purpose is to link the alumni with present students of the department and develop a network which is good for the professional and social development of the individuals associated with the department. And I know there are people in the Alumni Association who were students way back in the 60s. And I myself was a student in early 70s. But there are people who were even the department much before my time. So it's a good platform where we have brought the ex-students of the department together. And through various programs, we continue to be a link between the past and the present, and hopefully the future. Thank you, Pushpa.
Thank you, sir. We move on. Before I ask Professor Atrava to invite the speaker, the Department of Biochemistry has always had wonderful heads who have led the department. Since January 2020, we have Professor Ratnaprabha, who is MTech from IIT Kharagpur, PhD from IIT Bombay, and a leading protein biochemist as the head of our department. I request her to introduce the guest for today, Professor R.K. Rao.
Thank you, Pushpa, for your kind words of introduction. Honorable Dean, Faculty of Science, Professor Sarabhai Kataria, Professor CV Ramakrishnan, distinguished members of alumni, and invited guests, colleagues, and dear students. It has been a very happy occasion for all of us that we have gathered to celebrate Professor CV Ramakrishnan 96th birthday. Department of Biochemistry and Cell and Molecular Biology program, the fruits of his creative and scientific genius. His desire to interact with the students on his 96th birthday is more than an evidence for his dedication to science and education. On this very special occasion, I wish you a very happy birthday, sir, and look forward to many such occasions in future, where you will address the students, interact with them, guide them, and you would be the guiding force for their futures. Our alumni has always been a matter of pride for all of us. They have distinguished themselves in many different areas and proved to be true achievers. One such alumnus is Professor R.K. Rao. He did his PhD from Department of Biochemistry in the year 1987, and after that he did his postdoc from University of Arizona. Later, he moved to Medical University of South Carolina as assistant professor. In the year 2000, he moved to University of Tennessee as associate professor, and he is continuing there as professor. He has been working in the field of gastroenterology, and his work contributed a lot to our understanding of gut physiology. He is a member of many professional bodies and editorial boards of several journals. His work has always been funded by such prestigious organizations like NIH. He is also occupying several administrative positions in University of Tennessee. Besides, I must add that he is also known for his extracurricular talents. While he was a member of Biochemistry department, he was known as RK of Biochemistry department for his theatrics. This is a thing I learned, and I really felt very happy that a multifaceted personality like R.K. Rao belongs to our department. And another thing is, he has always maintained contact with the department, and whenever it was possible, he visited the department, interacted with the students, and also presented his work to all of us and enlightened us in a way, in a field where we are not working. So that way it was really kind of him and great of him. With these few words of introduction, I request Professor R.K. Rao to present his talk.
Sir, yeah. One minute, sir. Yeah, before R.K. Rao sir begins his talk, there's a small video the students have made for Professor CVR. You would like to present that?
Yes, I just want to add one line for the audience, that if you have any questions during the talk, please post them in the chat box. I request Dr. Ravi to handle the question answer session.
Yes, ma'am. I'm also along with it. I look at the YouTube channel. They will be posting there. I'll be asking questions here.
Okay.
Daria, please on the video. Daria.
Happy birthday.
Happy birthday.
Thank you very much.
Happy birthday to you, sir.
Birthday, happy
Thank you,
birthday.
thank
Happy
you,
birthday,
thank you
sir.
all. Thank you very much.
So we will now have the talk of Professor R.K. Rao. I request everybody to keep their audio off. There's a lot of disturbance. Please keep your audios off.
Yes.
My point is the present show, right? On the entire screen. You have to show the entire screen or just the PowerPoint.
Your voice is breaking.
Yes, sir. Show
Yes,
the
you are right.
entire screen.
Pardon me?
You can present an entire screen, sir.
Oh, I see. Okay. So let's see. Can you see?
Yes.
Yes, sir.
Okay. All right. Thank you very much for the invitation and the kind introduction. I am actually one of the fortunate to have the opportunity to be mentored by Professor CV Ramakrishnan for my PhD program. And it's an honor to present this seminar on his 96th birthday. And I hope to have the opportunity to do the same on his 100th birthday. So what I'm going to do today is to present to you some of our recent findings in understanding the mechanisms involved in alcohol-associated tissue injury at the gut-brain axis. And I hope at the end of the presentation, you can appreciate the conclusion that the gut is a primary target for developing treatments for the alcohol-associated diseases and disorders. 2018, the WHO fact sheet have identified nearly 200 alcohol-associated diseases, and at least 60 diseases are causally linked to alcohol consumption. And the good news is that approximately 20% of alcoholics don't develop any diseases, but the bad news is approximately 20% do develop alcoholic diseases targeting all different organ systems. So unfortunately, the behavioral intervention is not very effective in treating moderate and heavy drinkers, and there is no targeted therapy available right now to treat the alcohol-associated diseases. And for example, the alcoholic hepatitis right now is treated with the glucocorticoids, which is good for only short-term treatments and not good for long-term treatments. And many of these patients are resistant to glucocorticoids, and the mortality rate is greater than 40% in those that are resistant to glucocorticoids. So it is extremely essential to identify the novel therapeutic target to develop the therapeutic strategies to treat these diseases. So the precise mechanism currently is not known, precise mechanism that would explain the pathogenesis of alcohol-associated diseases. But there are three major denominators that kind of are common for many types of alcohol-associated diseases. One of them is dysbiosis gut microbiota, that the composition of gut microbiota is altered, mainly the increase in the pathobionts and decrease in the beneficial bacteria. Second is the gut barrier dysfunction. The barrier function means that the intestinal epithelium prevents the diffusion of any macromolecules such as allergens and toxins and pathogens to get into circulation. And that barrier function is disrupted in alcohol-associated diseases. And third is the endotoxemia. There is an elevated level of bacterial toxins in the blood. And So based on the current information, the prevailing hypothesis is that the alcohol kind of it caused the dysbiosis by changing the gut microbiota composition, therefore increasing the endotoxin level in the colonic lumen. And second, it disrupts the epithelial barrier. And once the epithelial barrier is disrupted, these endotoxins can absorb across the epithelium into the portal circulation, into the liver, and it can cause liver damage there. And then it also then spills into the systemic circulation, causing a condition called endotoxemia. And once you have the endotoxemia, it can develop a systemic inflammatory response. That means there's an increase in the level of pro-inflammatory cytokines in the blood. And once you have endotoxemia and systemic inflammation, then every organ is a target, and it can lead to multiple organ damage. So the barrier function of the epithelium is provided by what is known as tight junction. There's a tight junction is the junctional complex between the epithelial cells that kind of seals the space and prevents the diffusion of any macromolecules like toxins or allergens across the epithelium. And this is a multi-protein complex consisting of many transmembrane proteins, such as occludin, claudins, and the intracellular domain of these transmembrane proteins interact with many adaptor proteins like ZO1 and ZO2. And the adapter proteins in turn bind to numerous proteins, including the cytoskeletal protein, signaling proteins, and then this entire protein complex can interact with the actin cytoskeleton. So this is the transmission electron microscopic image showing the apical end of the epithelial cells. There are two cells here. This is the intracellular junction. You see the microvilli there. The electron dense area at the intracellular junction right at the apical end represent the tight junctions. And as you can see, the membranes from two cells are tightly occluded together, so there's not much space for diffusion of any macromolecules. And there's another junction complex below that is called Arran junction, which is not a barrier junction, but it has an indirectly kind of regulate the integrity of the tight junctions. Now, these tight junctions are kind of a circumferential ring around the cell. So if you stain for tight junction proteins like occludin and ZO1 by immunofluorescence method, and if you look at the fluorescence image from the top, it will look like it will give a chicken wire pattern of distribution by concentrated. These proteins are considered the intracellular junctions. So one of the experimental approach to study that is an in vitro model using a cell culture model of the intestinal epithelium, and we use K-coated cell monolayers. We grow them on a transwell inserts. These are the cups that they sit on either the 12-well plate or the 6-well plate or 24-well plates. And the bottom is made up of a polycarbonate membrane, which is semi-permeable because it has pores of different sizes. And
sorry to interrupt you, Professor Rao. There are few questions. Would you like to answer in between or in the end of the talk?
No, I can answer it
there
now.
There are few questions. Okay. Professor Park, can you unmute and tell your question?
Well, all I wondered was whether ethanol would ever reach large intestine. My understanding is that it would be absorbed much before that.
Yes, you are right. The most of the ethanol is absorbed in the stomach as well as in the proximal part of the small intestine. Doesn't reach to the lumen of the distal small intestine or the colon, but ethanol is kind of freely permeable to the membrane. So once it gets absorbed into circulation, then it can reach the colon. And basically, the ethanol concentration detected in the colonic lumen is same as the concentration of the plasma. So it's whatever present in the blood that can reach the colonic mucosa as well as the colonic lumen. And when it gets into the lumen, the bacteria can metabolize that one. So you are right about that. But that concentration is high enough to generate enough acetaldehyde, enough alcohol in the colonic mucosa can cause damage.
Okay.
What's another question?
Yeah, there's another question. Dr. Gore, you can go ahead and ask your question.
Sir,
So, Doctor,
sir, I worked in eastern Uttar Pradesh in a Gorakhpur area and all that. We had a huge enterovirus infections, encephalitis, and other things in that area because the water level is very high from the only 2 feet from the ground, and everything was getting contaminated. So, and they have our E. coli, coliform was everywhere. I mean, we will celebrate if we get a negative once in a while. And numbers are huge. WHO says 0, but we used to get 10 probably per 10 ml kind of things. So will that affect the entry of different viruses by damaging the this thing, intestine walls? That's what the thing, in a way, that's what you are actually telling me. I'm going to copy it.
Yeah, many pathogenic bacteria have been shown to disrupt the tight junctions. So like E. coli toxins or the cholera toxins and many other bacterial which are pathogenic. They release certain molecules, and they can kind of disrupt the junctions and then allow the entry of the bacteria, entry of the virus, of course, along with the toxins and in the allergen. So that is pretty much every intestinal diseases has been shown to be associated with the opening of the tight junction. As long as the tight junction is tight, and doesn't matter what is there in the intestinal lumen, it protects the body. So it is possible that any type of pathogenic bacteria can open the junctions.
Yeah, thanks, sir. Because there is enterovirus meningitis in USA also. So is it associated with epidemiologically? That study might be done. I mean, I do not know. So you can probably look at it.
Yeah. Okay. I'm not sure about that.
Yeah. I mean, anyhow, just I mean, whatever. Okay, sir. Thank you, sir.
All right. So this is one of the models that we use. This is in vitro model, as I mentioned. We can measure once we grow this monolayer to confluence and the tight junctions are formed, we can measure the barrier function by placing one electrode on the apical compartment, another electrode on the basal compartment. And if the tight junctions are tight, then you would see a very high electrical resistance across the epithelium. And if the junctions are disrupted, then you would see a drop in the resistance. We can also administer the extracellular markers like inulin, like fluorescently labeled inulin into the apical compartment. And after some time, you measure the fluorescence in the basal compartment, see how much fluorescence diffuse into the basal compartment. If the junctions are tight, you would see only a trace amount of fluorescence here. If the tight junctions are disrupted, then you would see increase in the fluorescence in the basal compartment. So you can also fix these monolayers and stain for any tight junction proteins and visualize the distribution of these tight junction proteins and gives an indicator of the tight junction integrity. We can also manipulate the epithelium genetically by either expressing certain proteins or downregulating certain proteins. So you can analyze the importance of certain molecules in the tight junction regulation as well. So our goal is we use this model to test the effect of alcohol on the intestinal epithelial barrier dysfunction. Now, the alcohol is known to be metabolized by alcohol dehydrogenase into acetaldehyde, and acetaldehyde is metabolized into acetate by aldehyde dehydrogenase. And this is the major alcohol metabolism that takes place in the liver. But the intestinal mucosa also express these enzymes to some extent, generate acetaldehyde and acetate there. And most importantly, in the microbiota in the colon, they express alcohol dehydrogenase, but very little of aldehyde dehydrogenase, and therefore the bacterial metabolism of alcohol leads to accumulation of acetaldehyde, which is nearly 100-fold more toxic than the ethanol. And based on our previous studies, the ethanol by itself doesn't open the junctions. It requires that ethanol is metabolized into acetaldehyde. And then in the numerous studies during the couple of decades, we show that the acetaldehyde can disrupt the junctions, and various different mechanisms that are involved. We have kind of identified that, published numerous papers to establish this mechanism. Basically, in the recent work, what we kind of focused on is that in vivo, you would see both ethanol and acetaldehyde. Is there a crosstalk between these two components? What happens if you have both of them? And this graph here shows the barrier function in the presence of ethanol and acetaldehyde. And we use the concentration, it's a suboptimal concentration, that ethanol and acetaldehyde alone had no significant effect on the permeability. But when we combine them together, we see a significant increase in the permeability in a time-dependent manner. And thus suggested that there's some sort of a synergy between the ethanol and acetaldehyde. This is kind of significant because most of the time you present some data with the, even for the grant or the manuscripts, they always say that your concentration is kind of, beyond the physiologic concentration. It's very easy to make that comment. However, it's very difficult to kind of demonstrate a physiologic concentration and physiological effects in vitro systems, because in vivo, it is not just one factor that is causing the damage. It's a multiple factor together. There is a synergy between the multiple factors. So this particular experiment kind of shows that when you combine them together, you need only a physiological concentration of ethanol and acetaldehyde to cause the damage. So obviously, our next question was, what's the mechanism involved here, right? So ethanol has been shown to increase the intracellular calcium in multiple types of cells. This has been shown for the last couple of decades. And so we wanted to know whether the ethanol and acetaldehyde increase the intracellular calcium. And what we found is that when we administered ethanol or acetaldehyde alone, and there was no significant change in the intracellular calcium. However, when we administered ethanol here, and then after 10 minutes, we added acetaldehyde, now you see a rapid increase in the intracellular calcium, reaching the plateau by about 15 minutes. So the next question obviously is, where is this calcium coming from? Is it calcium released from the ER within the cell, or is it a calcium influx from the extracellular medium? So we tested, evaluated the effect of a tapsicordine, which is an inhibitor of ER calcium release, or we also tested the effect of calcium-free medium. There is no calcium in the extracellular medium. So if the calcium influx is involved, then we would see that have an effect. And indeed, we found that the tapsicordine had no effect, suggesting that the release of ER calcium is not involved. On the other hand, calcium-free medium completely blocked the ethanol and acetaldehyde-induced calcium release, suggesting that there is a calcium influx is involved. Calcium is coming from the extracellular medium through certain calcium channels, obviously. So the question is then, is this calcium is involved in barrier dysfunction? So we measured the inulin permeability, found that the ethanol-induced increase in the inulin permeability was blocked by BAPTA, which is the intracellular calcium chelators. So therefore, it reduced the intracellular calcium. Tapsicordine had no effect, so ER calcium is not involved. Calcium-free medium almost completely block the effect of ethanol and acetaldehyde, suggesting that this increase in the intracellular calcium is involved in the disruption of the barrier function. And so this disruption of the barrier function is due to the disruption of the tight junctions. And so we stained these cell monolayers for occludin and ZO1. In control monolayers, you can see they are nicely localized at the intracellular junctions. And in the ethanol and acetaldehyde treatment disrupted these junctions. And this effect of ethanol and acetaldehyde could be blocked by BAPTA and calcium-free medium, but not effectively by tapsicordine, suggesting that the intracellular calcium-mediated disruption of the tight junction is involved in the ethanol and acetaldehyde-induced barrier dysfunction. One of the interesting observations we made here is that in the calcium-free medium, when we removed the calcium only from the apical compartment, we were able to block the effect. On the other hand, if we removed calcium only from the basal compartment, there was no effect, suggesting that the calcium influx from the apical membrane is involved. Certain calcium channels on the apical membrane is responsible for this increase in the intracellular calcium. So obviously, next question is that what type of calcium channels are present on the apical membrane of the intestinal epithelium? And it is very well established, two important channels that are present on the apical membrane of the intestine is the TRPV6 channel and the Ca1.3 channel. And this is localized on the apical membrane. And currently, until now, and this has been known for a long time, they have been studied this for a long time. And until now, we know that the function of these channels only in the calcium absorption, the calcium, dietary calcium, and the calcium from the lumen is absorbed through this TRPV6 channel and then Ca1.3 channel into the intracellular compartment. And then it is then pumped out across the basal arterial membrane into circulation, right? So our study, probably the data I'm going to present you next is the first one to show that these type of calcium channels on the apical membrane is also involved in some other functions or other, pathologic situation in the intestine. So the next question, my whole presentation will be focused on TRPV6 channel, and we have done studies with the Ca1.3 as well, but that's another entire different story. So here, what we did is that we knocked down the TRPV6 channel from the K-cortical monolayers by using the TRPV6 specific shRNA. As you can see, the TRPV6 channel protein is reduced without affecting the Ca1.3. And when you use the non-specific RNA transfected cell monolayer and the shRNA transfected cell monolayer, evaluated the effect of alcohol, ethanol, and acetaldehyde on the inulin permeability. As you can see in the red line represent the ethanol and acetaldehyde effect in the non-specific RNA transfected cell, and the purple line showing the effect in the shRNA transfected cell. You can see a dramatic reduction block in the effect of ethanol and acetaldehyde in the absence of TRPV6 channels, suggesting that it is important. So we also stain the cell monolayers for occludin and ZO1 or E-cadherin beta-catenin, which are the proteins of, excuse me, the adenine junctions. As you can see in the non-specific RNA transfected cells, the ethanol and acetaldehyde disrupted the tight junction as well as weakened the adenine junctions. But in the case of TRPV6 shRNA transfected cells, the junction, the tight junction proteins as well as the adenine junction proteins are intact at the intercellular junctions in the presence of ethanol and acetaldehyde, suggesting that the lack of or reduced expression of TRPV6 blocks the ethanol and acetaldehyde-induced disruption of the tight junction and adenine junctions. We then also wanted to test whether we could demonstrate the role of TRPV6 in the another model of the intestinal epithelium, that is the organoid model. So the K-cortical cell is a transformed cell monolayers, and it's also homogeneous type of cells, whereas the intestinal epithelium is known to have multiple types of cells. And so we grew the enteroids from basically we took a piece of the intestine and then isolated these crypts, which has the stem cells at the bottom. These isolated crypts were then grown on matrigel as a 3D model. And initially it forms a spheroid and then grows into an organoid. And these crypts also kind of divide, the stem cells divide, multiply, and then multiple crypts are formed. And this is a phase contrast image showing how the enteroid look like. And this is from small intestine. We can also grow the same from the large intestine. And we use these organoids to measure the barrier function and then the effect of the ethanol and acetaldehyde. Basically, we micro-injected ethanol and acetaldehyde into these organoids, intestinal organoids. And we also micro-injected the fluorescently labeled dextran. All these three are micro-injected into that. And after varying time point, we took fluorescent images of these organoids. Of course, we have this transgenic mice which express the GFP tagged occludin and RFP tagged ZO1. When we develop the organoids from those mice, and we can actually image these organoids, live organoids for the fluorescence, and we can see the tight junction proteins around around the epithelium. And so we, after certain time, if the junction is weaker and if the ethanol breaks the tight junctions, you will see the fluorescence from the lumen will diffuse into the extra organoid space. And so we measure the fluorescence from both compartments, and the outside and inside we present as the outside to inside ratio to evaluate the permeability of this epithelium. So here is the representative data. I show that you have the fluorescence, the dextran fluorescence inside. At various time points, we took the live organoid images, and then we calculated the fluorescence outside to inside ratio and plotted it against the time. As you can see, that outside to inside ratio kind of increased with the time, linearly, if not exponentially to some extent here. And that's suggesting that the ethanol kind of disrupt the barrier of the epithelium in a time-dependent manner. So we then grew the organoids, the intestinal organoids from the wild-type mice and the TRPV6 knockout mice. Did a similar study by micro-injected the ethanol and acetate. And then we took images at 10 minutes and 110 minutes. As you can see here, in the wild type, the fluorescence inside is decreased, whereas in the TRPV6 knockout mice, the fluorescence seems to be very similar, whether it's 10 minutes or 110 minutes. So when we measured the outside to inside ratio, it was clear that in the wild type organoid, the ethanol and acetate increased the permeability. In the TRPV6 knockout organoids, that effect of ethanol was completely blocked here. And we could also fix these organoids and stain for tight junction 13, so Cleveland 01. This is a control organoid, and the ethanol and the acetate disrupt the organization of these junction proteins in the wild type organoids, but not in the TRPV6 knockout organoid. So the next question was to know, instead of knocking down TRPV6, what happens if we just inhibit the TRPV6 channel, and can that also block the effect of ethanol and acetaldehyde? So we used the inhibitor called the SORC13, which is a TRPV6 specific inhibitor, and it doesn't inhibit the TRPV1 channel or TRPV5 channel. It only acts on the TRPV6 channel. And we evaluated the effect on the barrier function in cell monolayers. And here it shows that the ethanol and acetaldehyde decrease in the absence of inhibitor, decrease the resistance, increase the permeability. And in the presence of SORC13, there's a significant reduction of ethanol induced permeability, and that was reflected in the tight junction staining as well. As you can see, in the absence of inhibitor, you see the disruption of the junction. In the presence of inhibitor, this effect of ethanol on tight junction was blocked. We also evaluated the effect of SORC13 in the organoid model. As you can see, in the absence of SORC13, the fluorescence inside is decreased, but in the presence of SORC13, it was intact. And when we measure that one as an outside to inside ratio of the fluorescence, it is very clear that in the absence of inhibitor, there's a significant increase in the intestinal permeability, whereas the epithelial permeability, and that was completely blocked by SORC13. And this kind of confirms the role of the TRPV6 channel in the ethanol and acetaldehyde-induced permeability. So in the next question, we wanted to know whether TRPV6 role in the ethanol-induced gut permeability, the disruption of the junction and gut permeability, can we establish that in an in vivo model? So we used a wild-type mouse and the TRPV6 knockout mice, and we used a chronic alcohol administration model in these mice, which is a 4-week-long experiment. We feed the mice with the alcohol in a liquid diet called Lieber-DeCarli liquid diet, very well established liquid diet for the alcohol treatment. And we increased the alcohol concentration on stepwise manner from 2% to all the way to 6% at the end of 4 weeks. And then we measured the gut permeability, mucosal permeability, by measuring the vascular to luminal flux of fitzinylin. Basically, we inject fitzinylin but we are tail vein injection, and then collect the intestinal flushings after 1 hour and see how much of fluorescence is leaked into the gut lumen, and as an indicator of the barrier dysfunction. Tight junction integrity was evaluated by confocal immunofluorescence microscopy for tight junction proteins. We take the cryo sections of the colon or the intestine and then stain it by immunofluorescence method and perform confocal microscopy to visualize these proteins. So, we measure the plasma LPS level as an indicator of endotoxemia and measure the plasma cytokines by ELISA as an indicator of systemic inflammation. And the liver damage was analyzed by measuring the ALT, AST levels in the plasma, which is a clinical indicator for liver damage, and also measure the liver triglyceride level to assess the steatosis, alcohol-induced steatosis. And the hepatitis was analyzed by measuring the inflammatory cytokine mRNA expression. And the neuroinflammation was also analyzed by measuring the mRNA for pro-inflammatory cytokines in the cerebral cortex. I'm presenting only the representative data to, in the interest of the time. And the inulin permeability in vivo in this mice showed, in the wild type mice showed a significant increase in the inulin permeability, suggesting that the barrier function of the intestinal epithelium is disrupted in the wild type mice by the alcohol feeding, but it was absent in the TRPV6 knockout mice, suggesting that the TRPV6 is involved in this process. And when we stain the prior sections of the colon here in the wild type mice, this is in the pair-fed mice, the junctions are intact because of fluids you want to localize at the intercellular junctions of the epithelium, and it is dramatically disrupted in the ethanol-fed mice. But in TRPV6 knockout mice, the junction seems to be intact even in the ethanol-fed mice, suggesting that the absence of TRPV6 completely blocks the alcohol-induced disruption of the tight junction and barrier dysfunction. The plasma LPS level, again, was increased in the wild-type mice by the alcohol feeding, and this effect was absent in the TRPV6 knockout mice. And similarly, the plasma TNF-alpha or the MCP1 levels were increased by alcohol feeding in the wild-type mice, but not in the TRPV6 knockout mice. Here's the representative data for regarding the liver damage. The AST levels or plasma ALT levels higher in ethanol-fed wild-type mice, which was absent in knockout, TRPV6 knockout mice. And this is the histopathology showing in the wild-type mice, alcohol feeding induces histopathological changes. These white dots, what you're seeing is predominantly the accumulation of fat droplets in the liver. And in TRPV6 knockout mice, the liver histology showed very similar to the pair-fed mice, very normal appearance here. And the liver triglyceride levels were increased by ethanol feeding in the wild-type mice, but not in the TRPV6 knockout mice. And also, when we measured the mRNA levels for pro-inflammatory cytokines like IL-1 beta or the chemokine CCL5, again, they all show that the ethanol increased the expression of these cytokines in the wild-type mice, but not in the TRPV6 knockout mice. Here is the representative data on the neuroinflammation, measuring the TNF-alpha mRNA levels in the cerebral cortex, or the TNF-alpha, MCP1, and TORC-B. TORC-B is an anti-inflammatory factor, whereas TNF-alpha and MCP1 are the pro-inflammatory. As you can see, the alcohol increased the level of mRNA for TNF-alpha and MCP1 in the wild-type mice, but not in the knockout mice. And the opposite, you could see that the TORC-B mRNA levels are reduced, which is, so it reduces the anti-inflammatory factor, and that was also absent in knockout mice, suggesting that the TRPV6 channel is involved. And our hypothesis that it is the TRPV6 channel that is present at the intestinal epithelium is causing all these differences in the neuroinflammation and liver damage. So then we asked the big question, how does ethanol activate tryptase-6 channel? So to answer this question, we measured the ionic currents in the K-cortisol monolayer by a whole cell patch clamping, and then evaluated the effect of ethanol and acetaldehyde ionic currents with or without the presence of source C13, which is the tryptase-6 channel inhibitor. So here is the voltage current clamp, voltage current plot showing the effect of EGTA, ethanol, acetaldehyde, and ethanol plus acetaldehyde. The EGTA is a well-known activator of tryptase-6 channel, so we use that one as a positive control, and we can see the EGTA, and the data is summarized here. We kind of normalized all the effect of ethanol, acetaldehyde, normalized its response to EGTA because it's a positive control. Ethanol at 50 millimolar had a very little ionic current, elicited very little current. But if we increase the concentration of 150 millimolar, now you see a significant increase in the ionic currents in K-cortisol monolayer. Acetaldehyde at 200 micromolar also elicited significant ionic currents in K-cortisols. And when we combine this 50 millimolar ethanol and the 200 micromolar acetaldehyde together, now you see a dramatic increase in the ionic currents. It's almost pretty close to the EGTA current here, and suggesting that there is a synergistic activation of tryptase-6 channel by the presence of ethanol and acetaldehyde. And of course, this is a whole cell patch clamp. We don't know what the ionic current is caused by or due to which type of channel. So for that reason, we evaluated the effect of source C13, and you can see that the source C13 completely blocked these ionic currents elicited by ethanol and acetaldehyde, suggesting that tryptase-6 channel is involved in these ionic currents induced by ethanol and acetaldehyde. So we also conducted these studies in hex cells by expressing tryptase-6. The hex cells do not express tryptase-6 channel, but when we transfected these cells with the tryptase-6 and we measured the ionic currents in response to ethanol and acetaldehyde and EGTA in these cells, so now it kind of gives us a more direct effect of ethanol and acetaldehyde on tryptase-6 channel. And here, this is the real-time changes in the ionic currents in response to EGTA, ethanol, and acetaldehyde. As you can see, the data summarized here, the ethanol induces a very strong increase in the ionic currents in these hex cells expressing tryptase-6 channel, and acetaldehyde also elicited ionic currents in these cells, and we could actually block these effects by source C13 by demonstrating the effect is caused by the tryptase-6 channel. And instead of tryptase-6 channel, when we expressed hex cells with the TRPV1 channel, the ethanol, acetaldehyde, or EGTA had no significant increase in the ionic currents. On the other hand, capsaicin, which is TRPV1 is a receptor for capsaicin. Capsaicin has a strong activator of TRPV1, and you can see that capsaicin did increase the ionic currents in these cells that express TRPV1 channel, and therefore suggesting that the ethanol and acetaldehyde effects are very specific for tryptase-6 channel and no effect on TRPV1 channel. So our question then is that because of this effect in the hex cells, we asked the question, does ethanol directly bind to tryptase-6 channel? So how is it activating? So to answer this question, we prepared the recombinant human tryptase-6 and incubated with az-butanol. The az-butanol is a photoactivatable butanol, and when you photoactivate it, this butanol will form a crosslink with any of the region where it binds to it. So we cross-linked the butanol to tryptase-6 channel by photoactivation, and then did a proteomic analysis by trypsin digestion and LC-MS analysis, and we found that there was one site where butanol was able to bind, that was histidine 225 in human tryptase-6 channel. And this histidine 225 in human TRPC corresponds to histidine 185 in the rat TRPC channel. There is a 40 amino acid difference between these two species at the end terminus. So basically, if you look at the structure of the TRPC channel, this is located, the histidine 185 in this rat TRPC is located right in this region, which is the N-terminal end of the molecule. And this is the actually a anchoring rich repeat domains, which where it's pretty much a regulatory domain. The calcium calmodulin complex binds to this and regulates the activity of the TRPC channel. And so it's interesting that this particular butanol binding site was present in this anchoring repeat domain. And we also did a computation analysis to identify any other ethanol binding sites, known ethanol binding amino acids surrounding this histidine 185. We found one of the closest one is the arginine 134. So what we did is that we induced mutation to this histidine 185, and also we induced mutation to histidine and arginine as a double mutant, and then transfected that one into hexels, and then measure the ionic currents in response to ethanol and acetaldehyde. So here is the plot, the current to the voltage plot here in wild type, the histidine 185 to alanine mutant, and the arginine and histidine double mutant here. And this is the real-time changes in the ionic currents. And as you can see, this is the wild type current in response to ethanol. And this is the in the single mutant, and here's the double mutant. When you summarize this data, it shows that compared to wild type TRPC channel, the ethanol response is significantly reduced in the single mutant, that is histidine 185 to alanine mutant, and the double mutant, you see a further decrease in the ethanol response, suggesting that this may be an ethanol binding pocket. Maybe there are a few more amino acids are involved forming this ethanol binding pocket, and that when it binds to the TRPC, it kind of activates the TRPC channel. So to summarize that, the TRPC channel is involved in the calcium absorption and through the apical membrane into the intercellular compartment, and then is pumped out across by the calcium ATPase across the basolateral membrane into circulation. But the key fact here, important thing here is that, and this calcium absorption has to occur without changing the intercellular calcium. If you increase the intercellular calcium, then it's going to change the function of the cell. So the calcium absorption takes place at a very high concentration without changing the intercellular calcium. The entire mechanism involved in how it does without changing the intercellular calcium is not clear, but there are some theories suggesting that one, the intestinal epithelium express cal binding, they can bind calcium and therefore reduce the intercellular free calcium. And it also, there is a feedback inhibitory mechanisms has been suggested that the calcium calmodulin complex can bind to TRPC channels and temporarily inhibit the channel. So therefore, repeated activation and inhibition can keep the intercellular calcium low. And alcohol, for some reason, using this TRPC channel, induces the calcium influx, but in this case, you see an increase in the intercellular calcium, suggesting that the alcohol is somehow either modulates the TRPC channel so that it cannot be inhibited, or it alters some other mechanisms within the cell, and therefore it increases the intercellular calcium, and that is required for opening of the tight junctions. So the ethanol and acetaldehyde can bind directly to TRPC channel, increase the intercellular calcium, and that results in opening of the tight junction. And I put a black box here because it is likely that there is a cascade of signaling pathway that is involved in the calcium, between the calcium and the tight junction disruption. Based on the data that we have, some of the mechanisms in this black box include the oxidative stress, CSARC activation, and tyrosine phosphorylation of tight junction proteins, particularly the occludin and loss of protein-protein interaction leading to tight junction disruption. When the tight junction is disrupted, the LPS from the altered gut microbiota here, it gets into the circulation, cause systemic inflammation, and cause multiple organ damage. And particularly the data that we have presented show the liver damage and neuroinflammation depends on this TRPV6 channel mediated intercellular calcium and LPS absorption. So with that, I would like to acknowledge our lab members, past and the present, who worked in our alcohol project. I just want to highlight two students who came from the program. Shunali and Kesha came from the integrated cell biology and molecular biology master's program. And we will probably have another student this year. And department collaborators, Cordero Morales, Valeria Vasquez, and Briar Bell, and Rebecca. This is the team we collaborate with to measure the ionic, the ion channel activities. And then John Jager and his group, we collaborated for calcium imaging, and other collaborators in our institute, Ankush Gosain, who's an expert in microbiota. And we study the microbiota in the alcohol model, and I have not addressed that one. That's an entirely another story which we are working on. And Joe Pierre is also a microbiota expert, in our institution. I collaborate with Georgiani, who is an expert in mass spectrometry. So all of our studies with the structure and identification of the sites and phosphorylation sites and the ethanol binding sites were done in collaboration with Francesco. And we have other collaborators, University of Cincinnati, Chip Montrose and Ihara. They were involved in they introduced us to growing the enteroids and organoids. Laura Nagy provides from the Cleveland Clinic, provides us with the clinical samples. And Jerry Turner from the Harvard is a good friend of mine for a long time. We collaborate and exchange information about the tight junction structures. And we have some collaboration in Germany particularly, and they have been very generous in providing us with various types of transgenic, in the conditional transgenic mice, etc. And finally, I would like to thank acknowledge the funding by NIH from both NIAAA and NIDDK. We just really got renewed this NIAAA grant for another 5 years. And the VA Merit Award, we just got this renewed as well. And UTHSC support grants. So with that, I'd be happy to take any questions.
Thank you, Professor Rao. That was indeed very wonderful talk and very interesting. So now there are two questions which are there in the chat box, and others can also ask. Students will be asking in the YouTube channel, and I'll be putting forward their questions. So Professor Naresh Kumar is asking, can low level of alcohol regular consumption contribute to intestinal damage? Low level consumption on regular basis.
Low level of, the NIAAA has been kind of addressing this. What is the level of alcohol that you consume is safe? Because there is also, to some extent, there is some data to show that the small amount of alcohol may be good for you. Like French drink a glass of wine, every day, and that they do not have any issues, any health issues related to alcohol. And so, according to NIH, they say one glass of one drink per day for women and 2 drinks per day for men are just fine. So that's the so up to about 7 to 14 drinks per week is. And more other issue is that rather than this chronic drink, it is the binge drinking is a lot more. How do I get back to the meeting?
Yeah.
I'm thinking.
Yes, you can stop sharing. Yeah.
Yeah. Okay.
Stop sharing. The blue button. Yeah. Blue button. Yeah.
All right. So I can see you guys.
Yes. So second part of his question is like, how much damage is prevalent in the population? So are there any population studies on alcohol-induced damage?
Well, the population studies we have seen in depending on the expression of alcohol metabolic enzymes. For example, alcohol dehydrogenase or alcohol aldehyde dehydrogenase. In Asian population, they have this mutant aldehyde dehydrogenase in ALDH2 mutation, which makes it inactive. That results in an increase in the acetaldehyde accumulation. So therefore, many of the Japanese population people, they cannot drink well. If they are homozygous for this mutation, they cannot drink at all. If they are heterozygous, they do drink, but you can see the flushing in their face because of the high acetaldehyde concentration. And they are also a little bit more prone to develop the acetaldehyde, alcohol, mediated tissue injury. So like acetaldehyde is also a carcinogen. The alcohol and acetaldehyde can increase the risk of different types of cancers, including colon cancer, breast cancer, and these subpopulation with this mutant ALDH2, they are much higher risk to develop these cancers if they are drinking.
So one student, Shivani, has a question that is a particular time in a day will help it in a sense that trigemination might be clock regulated, circadian rhythm regulated. So drinking alcohol, consuming alcohol in a particular time in 24 hours may have different effects.
Mm-hmm.
It's a question from student.
I see. Well, I don't know if anybody has really looked at that, drinking in the morning or evening, whether have an effect or not. But whether it is in the human subjects or in the experimental model, anytime if the blood alcohol concentration is high, that's going to have an effect very similarly. So in mice, when we feed alcohol in a diet, and alcohol concentration much higher in the early morning because the mice have the habit of eating in the dark cycle mode. So we see that the blood alcohol concentration is also high in the early morning, and you can see that the intestinal permeability, endotoxemia also kind of correlates with the blood alcohol concentration. But in terms of extent of tissue damage, depending on which time you take, probably not likely, because it all depends on how much is the alcohol, what is the alcohol concentration in the blood.
But I had a question that, can I ask Ravi?
Yes, sure, ma'am. Go ahead.
Yeah. No, in case of wine and all, since it's mostly red wine, so you will have antioxidants, resveratrol, and things like that. And so, in contrast to that, in India, most of the people really don't drink wine. Low-income group will be drinking something else. So, in such cases, the antioxidant effect itself will moderate in case of French and many other populations.
So mostly the French drink more red wines, which has more antioxidants, and it was a kind of a famous as a French paradox, who consume alcohol every day, but they don't have much health issues. And antioxidant is definitely one protective factor there, and some have shown just even the alcohol at a very low concentration can also be protective. It's not all that, cardiovascular protection has been demonstrated. And, another thing is that most of these alcohol drinks also have very high level of acetaldehyde. The beer, any type of alcohol, the wine, they have the acetaldehyde. In fact, it's surprising that they are present in a millimolar concentration, and they themselves can cause damage, particularly the oral cancer and oral mucosal damage. Finland group have tried to show that the high acetaldehyde concentration in these drinks can cause damage right away.
So I have a couple of questions for you. One is, you say that the best effect is seen when ethanol and acetaldehyde are present together. Okay, so do you think there can be, like you showed, two residues which are target of ethanol? So there can be some amino acid residues which might have target acetaldehyde might have target.
Yes, the acetaldehyde may have another target. Acetaldehyde is known to form a shift-base adduct with the epsilon amino group of lysine. And it has been shown in many different types of channels, the enzymes, when they bind to it, they kind of inactivate the enzymes or inactivate the channel. The function is gone. And it is likely here that acetaldehyde may be binding to certain lysine amino groups in the tryptophan channel. That is our next step to kind of do a mass spec analysis. We can actually bind acetaldehyde and cross-link it with the sodium borohydride and then do a mass spec analysis to identify the site. And we're going to do the similar studies and to identify how exactly acetaldehyde, is involved. And then, as I mentioned, there's another story about the Cav1.3 channel. When we knocked out Cav1.3 channel or used the Cav1.3 knockout mice, we were able to block the ethanol effect. And we think that there is a crosstalk between these two calcium channels, the Cav1.3 and tryptophan. And then it looks like the acetaldehyde involves this Cav1.3 channel as well. So that's another story we're working on. And that's the calcium, that's a calcium, that's a voltage gated calcium channel. And, we did a preliminary study using the calcium channel blockers like the diltiazem, which is the, we take for hypertension in the clinic, and diltiazem could block, we were able to show that diltiazem blocked the ethanol effect.
Okay.
In case, go ahead.
Yeah, go ahead.
In case of humans, then inhibiting TRPVX is a possibility, or will it also affect the calcium absorption and therefore calcium status?
Yeah, that's a very good question, because that question was asked by a reviewer when we submitted a grant application. And for that reason, we did these preliminary studies by feeding the mice with the diltiazem for inhibiting the calcium channels, to inhibit the calcium channels. And we measured the blood pressure in these mice. Surprisingly, those the channel inhibitor blocked the ethanol effect, but there was no effect on the blood pressure. I think these channel blockers are effective in the hypertensive condition. On the normal condition, you don't see that, because the normal homeostatic mechanism is intact.
Any other question?
Yes. Can I ask that in certain circumstances, alcohol can be utilized as a food? Why I'm asking, because in 1964-65, I was working very closely with Professor Rajalakshmi, and she did a lot of such studies on the metabolism, and in the working class people, she found that the calorie intake was lower than the expenditure. And only way I think she was able to conclude that alcohol may be utilized as a food by these workers. So there are enzymes which can be induced by alcohol consumptions, and people can utilize them as a food.
Yeah, I would think so, because it's a very high calorie molecule. It can produce very high amount of calorie, because when it metabolized by alcohol dehydrogenase, it produces one molecule of NADH, and then when it's converted into acetate, another NADH molecule. So you can get two NADH molecules that can produce a significant amount of ATP, and it's also a quick generation of energy production. Very rapidly increases the energy level. So definitely it can add to calorie intake. Definitely it can conduct. So.
Thank you very much.
Any other question?
Not a question, but can I ask Dr. Rao whether you want to tell us, just maybe in a line, any significant effect on microflora, because this will be very, very relevant, for
In fact, that is another entirely different story that we are working on. Now we are getting it funded by VA for the next 4 years. We found that the alcohol, of course, it is known for a long time, the alcohol causes dysbiosis. It, changes the microbiota composition. But what we found is that there is a panel cell right at the crypt cell of the intestine, which produces an antibiotic, antibiotic peptides called alpha defensins. And these defensins, they are secreted to the intestinal lumen, and they kill the pathogenic bacteria, but they don't affect the probiotics or beneficial bacteria. And therefore, under normal physiological condition, these alpha defensins play an important role in maintaining this, normal microbiota composition. So what we found is that alcohol dramatically reduces the gene expression for all of these alpha defensins, and then we think that that is one of the mechanisms how it is inducing or altering the gut microbiota composition. And we also kind of synthesize this peptide, human defensin 5, which is one of the alpha defensin in the human, and then we feed that one to mice, and we kind of correct the gut microbiota. We can block the alcohol-induced damage to the intestine.
Wonderful. Yeah, very good. Yeah.
That's another story we're working on.
Yeah.
So,
Ravi, can I ask?
Yes, sure. Go ahead, sir.
Yeah, I want to flash the question which was posted by Dr. Milind Gore. Do we know anything about the composition of desi liquor that is highly consumed in India, and the composition and perhaps the effect of this?
I do not know much about the difference in the. I know that this is done by Finland group. They have been comparing all different types of alcoholic beverages, and trying to. The composition wise is, I think, you have the alcohol and you have other components like just like we are talking about the antioxidants and other components. And another thing, the Finland group was focusing on the acetaldehyde. So I don't know if different liquors in India have been. I'm sure maybe there is some data out there. I don't know about it.
Because I know people that class, labor class, they consume a lot and they consume every day. And I see them working.
Yeah. As I said, it's actually a very small subpopulation who really get affected by this. As I mentioned, 80% of the alcoholics, who drink alcohol every day, they're just fine. But it's only about 15 to 20% alcoholics who get into the health issues. And we are trying to address that. We think that now, trying to address why so, and we think that it could be the multiple hit model. It's like alcohol and obesity, or alcohol and hepatitis C virus infection, things like that. And one of the projects we are working on is the alcohol and the stress. We find that when you stress the mice, either holding them, restrain stress every day, and then give them alcohol, we see a very strong synergistic damage to intestinal mucosa, endotoxemia, and systemic inflammation. And we could actually mimic that by, instead of stress, even if you give just a corticosterone on a daily administration of corticosterone, we can reproduce that effect. We think that the glucocorticoid receptor mediated effect is, can actually increase the alcohol mediated damage. And that's probably one of the reasons why the alcoholic hepatitis, the glucocorticoid doesn't work all that well, because we find that it is actually probably causing the opposite effect.
Can I crack a joke here?
Yes.
I think people like Professor R.K. Rao will support in a great manner the DMK government in India, in Madras state, because they are promoting more and more liquor shops. Just a joke, I said. Because I have forgotten completely the spelling of biochemistry. since all these big people are asking you fantastic questions, then since I'm taking part in it, I thought maybe I should ask some questions. Not questions, some comment. That's why I gave this comment.
But sir, Gujarat is supposed to be a dry area, but the amount of liquor which is consumed here may be very similar to what is consumed in Tamil Nadu, illegally.
And we can also kind of make a joke of it. Every year we meet in an alcohol research society meeting here in the United States in different locations. We make a joke that most of the alcohol is consumed in the alcohol research society meeting, reception and banquet and so forth.
So there is a fun question from a student that why alcohol is consumed more in colder regions of the world.
Calorie, maybe increase providing the more calories so you can keep your body much more warmer actually. So.
And they also won't feel the cold when they go on drinking.
Yeah. They don't realize it.
Lose the cells.
So this another question I had on like leaky gut is a very common phenomenon, a fancy term nowadays. So leaky gut, which is not because of the alcohol consumption, will also be can be treated in the same fashion, or it has a different mechanism. Non-alcohol induced leaky gut.
Yeah, I think there are different mechanisms. For example, the cholera toxin or E. coli infection, pathogenic E. coli infection, they have a different mechanism disrupting the junctions, and people are trying to understand how to, control that. One of the mechanism people, which is more generic way to control the barrier function is, consuming probiotics. So that way you kind of probiotics not only kind of correct the microbiota composition, but it may, we have studied, we are looking at lactobacillus species, and we can show even Salmonella, they can directly affect the epithelial cells and, keep the junctions tighter. And we think that it is multiple mechanisms are involved. One of them is the activating the EGF receptor on the epithelial cell, and then tightens the junctions by tightening the actin cytoskeleton. And there's another species worked by different mechanism using the PKC mediated signaling. So that's some other mechanism. And of course, the others would be to more specifically kind of a targeted inhibition of barrier disruption.
One more comment is the alcohol consumption. Some individuals obesity and some people are thin. So what is the relation between alcohol consumption and physiology?
In obesity, you mean crosstalk between obesity and alcohol, or?
Alcohol, some people who consume alcohol, they become obese. Many of the people in India, you don't find them obese.
I think it's probably the genetic makeup. In mouse model also, there are multiple various different species. It depends how, some mice are more sensitive to alcohol, some are not. And for that reason, when we do an experiment, we always have to use, C57 BL6 mice. They're a lot more sensitive. You can see the body weight gain in those mice. And some other species, when we use the transgenic mice, we notice that there's no much change in the body weight gain. I think it's probably the genetic makeup. It could be probably a more complex situation, I think.
But the lean people also have some sort of a metabolic disorders. It is not that they are healthy. So there is some sort of a paradox here.
Yeah, I would think that, as I mentioned, probably the multiple hit models are the most important model. That's why we see so much of variation among the subpopulation of the human population. So some are more sensitive, some are less sensitive, some may generate, develop obesity, some not. So I guess it's all the genetic makeup. So I guess, as we, you probably all know that, eventually we're all leading into the, the more modern, individualized therapeutic system, right? So depending on your genetic makeup, they will prescribe you what type of medication is better for you.
Sir, now one of the huge in immunology, microbiome and immune response is studied. I mean, you can get a huge number of papers, and they add the stress and microbiome and immune response together, and that is what is combined of everything is together is affecting the disease, including the autoimmunity or cancer or other infections and everything. I mean, that is what is happening now. And that is a big grants on that, most probably.
Yeah, I mean, the immune system is obviously is one of the the highlighted area, and particularly the mucosal immunity is has been studied for a long time. And alcohol is a strong suppressor of immune system. So that's probably one of the mechanisms how the alcohol can cause health issues. It's not much work done in the mucosal immunity and the alcohol, but as you mentioned, it's a major issue. I think that needs to be studied in a lot more detail, and particularly during the current situation, it has been kind of known for a while that the chronic inflammatory diseases are very high in developed countries compared to the underdeveloped countries because of the maintenance of very hygiene. And I always wonder, during this last 2 years of pandemic, everybody's so clean, so everything needs to be kept clean. You're wearing masks. I don't know what is going to be the long-term effect. You probably will be further suppressing the immune system, not stimulating the immune system. So that's it's a very huge general question, and something. Yeah, it's an important question.
Now, all the infections which are preventing now because of the mask will come once we remove the mask, and we will get a huge.
It is going.
I mean, that's what will happen. I'm seeing that in my grandson now. To 1.5 years, he was not going the crash. Now he's going and he's getting infected with different kind of common cold and etc. So that's what happens. So that is what we are going to face it.
Looks like it.
Right, maybe.
I think we should stop here.
Yeah, I have one question. This is regarding how it influences the enteric nervous system and in which way it is connected with the gut-brain axis, particularly in the inflammatory scenario.
Right. Yeah. You're talking about. Did you mention in the beginning enteric nervous system, right?
Enteric nervous system. Yeah.
So there is no much work done in terms of how the alcohol affects the enteric nervous system. I think this is an important area. I strongly believe that enteric nervous system is affected by the alcohol consumption, because the enteric nervous system kind of regulates many of the intestinal functions, including the absorption, secretion, and motility, and all those things. And there is only one study I have seen. They're trying to analyze the gut motility, and TRPV1 channel is involved in the enteric system. It's expressed in the enteric nervous system. There was one study previously I've seen that they show that TRPV1 channel is affected by alcohol consumption. So I'm guessing that is expressed only in the enteric nervous system, and therefore the enteric nervous system function is definitely altered in these conditions. So my collaborator, Ash Gosain, who's a pediatric surgeon, we collaborate, we have our lab meeting together most of the time because he's working with the Hirschsprung disease. He's an expert in the enteric nervous system. He's planning to get into that and see how it works. It's definitely an important area. For a long time, it has been hard to kind of study that, but now they have new technologies to study the enteric nervous system in a lot more meaningful way. Yeah.
Thank you.
Yeah, I think we should.
Yes, I would like to thank Professor Rao. It was a wonderful talk, and you could answer most of the questions. There are some questions we will ask students to email you directly and get the answers from you. Thank you.
Thank you so much. So our main purpose was to celebrate Professor CV Ramakrishnan sir's birthday. I would now like to ask Professor Sarita Gupta to say a few words about the role CVR sir played in setting up the CMB Institute. So Professor CVR has got very good credit of playing a role in starting 2 institutions in MSU. And both are doing very well, the Biochemistry Department and the Cell and Molecular Biology Institute. So I would like Professor Sarita Gupta, who worked very closely with Professor CVR in setting up the institute, to say a few words before we ask CVR to speak.
Very CVR, though I can't see him anymore.
He will come, he will come.
No, he is there. He's
Okay.
right there.
Yeah, I also wanted to just say one thing, that there are many old students here whom he had taught, and all didn't get introduced to him. So that will be something that we probably should do. Some of us know him, but then others are here whom he didn't get to meet. So just in case, yeah.
So shall we do that first or later?
No, you go ahead and tell about Professor first. I mean this, yeah.
So good morning, good evening, and good afternoon, as I said earlier also, to everyone being here. I'm very happy that we are here to celebrate on virtual mode the birthday of Professor CV Ramakrishnan. I have worked with him closely since last 10 years. When we started in 2009, when Professor Venki got the Nobel Prize, after that we started working on this project. When Professor Ramesh Goyal, who was then the Vice Chancellor, thought about starting something to honor, because Venki Ramakrishnan is our own alumni of MS University, so he wanted that we should have something, and he discussed this matter with Professor CV Ramakrishnan, and I think it is his vision and the kind effort of Professor CV Ramakrishnan as well as Professor R.K. Goyal that we went to government of Gujarat talking about that can we start some new interdisciplinary course in life science. And it was, I think, Professor CVR's presence, and our Chancellor was also with us at that time, that gave a real boost. And then government of Gujarat although gave 1 crore of rupees for us to start the cell and molecular biology program, but simultaneously we were also working to get some money from DBT to start some interdisciplinary research and development of the infrastructure. And along with that, we had put to the DBT that we can start a new course in the line of cell and molecular biology, which is a 5-year integrated program. And we were fortunate at that time that we could get 13 crores from DBT. And because of these 2 grants which we got, we could start this cell and molecular biology program. And after 3 years, that means 2012, we can start the program. And for from 2009 till 2012, we have been working on this. And Professor CV Ramakrishnan was continuously supporting us, and then going to different institutes in the like TIFR, NCBS, ISER Pune. These are the important institutes where we visited and we tried to discuss the matter. And Professor Vijay Raghavan, who was at that time the DBT secretary, also was involved in this, and Professor Shashidhara, who was from ISER Pune. These are few names, like I cannot think about starting the cell and molecular biology program without their efforts. And behind everybody, it was only and only Professor CV Ramakrishnan who was the backbone for us, who was the true visionary and the true instrumental in starting the program of cell and molecular biology. I think this has been, I have been telling this every year to our students who are every year joining, but since last 2 years, because of the pandemic, it was like it was all virtual. And I am also very happy that the students from last 2-3 years who could not meet Professor CV Ramakrishnan because he was the one who used to visit us every year. And almost every year he was with us since 2012 onwards. And we were lucky to have interaction with him, the students, the faculty members, the vice chancellor, the dean, and everyone. And that is, the students were really very happy to have him. And then he was the inspiration to all of them, a motivating force, which has really built a very good scientific aptitude in these students. And it was unfortunate that since last 3 years, after 2019, he could not visit us. And the pandemic has not given any chance for these students to really interact with him. So I think this is a good opportunity when Professor CV Ramakrishnan can address them, and they will be able to listen to him and then interact with him. And that is one of the reasons that we thought about that this is a good way to start and to have a good interaction with the students. Just I want to tell the students who are here that it was, you must be highly inspired to know that Professor CV Ramakrishnan started the biochemistry department at the age of 24 years. So that is something which is really not easy for anyone to start at that age. And after that, he was here doing many things, and he is the one who has worked on carbohydrate metabolism and Krebs cycle, and he worked with like his thesis has gone to Krebs, and then he was very happy with sir, and then I think he has been invited there, and he worked with this Professor Krebs. So these are a few things. I don't have much time to talk about what has been done, but one thing is biochemistry department was known at that time when I joined in 86. It was only one meeting I had with professor in his room, and then he retired after that. So, but of course, I had good time with Professor Rajlakshmi because after that I was there for, and I then became the like I have got the faculty position in our department. But that was the only one meeting I had with. But I'm very happy that after that, if from 2009 onwards, I'm closely working with him, and it's real fortune for me to work with such a visionary, and I learned many things from him. And not only me, I think many of our students also learned a lot from him as he has been coming every year and interacting with the students. I just want to mention few things which he has done for cell and molecular biology. The first and the most important one is he made a very good licensing between all the big institutes in India, and that has made a very good way for the students to learn, interact, to do summer training at these places, and to go for dissertation, doing the dissertation in this. And I'm happy that this last 10 years, our MSc dissertation students could actually get some publications from their MSc dissertation work. And I must congratulate Professor R.K. Rao for that, that two students who had worked with him, they could get 3 or 4 papers from the work which they have done with him. So this is something which is really remarkable, because getting publications from the MSc dissertation is something very difficult. And it is possible because of the people who are around us and who has helped us, and it is the Professor CV, and all credit goes to him, because it is his efforts that we could interact with every institute, with the different professors, with different intellectuals, and thereby our students are getting benefited out of it. The other thing I would like to say about Professor CV, of course, a lot of things are there, but another very important thing is the donation which he could get for our building, where we could like, because of the donation, and I must thank Dr. Amrish Patel, he is here, that it is his donation and many others who are there who has donated for the cause, that we could develop a felicitation center for our CMB student, and it was very well developed, and we have actually inaugurated that. We wanted to inaugurate in presence of Professor CV Ramakrishnan, but unfortunately at that time it was not ready, and we had to wait for some more months, and then we inaugurated it. And Professor M.S. Patel was there with us at that particular time, and we had a lot of other, like for students have done a lot of other things, and competitions and other things have been organized. The other important point I just want to make is we have celebrated Professor CVR's birthday thrice at least, if I remember correctly. And 2019 was the last one when we celebrated here in Baroda. And at that time, as I said, that we could not have this building ready by then. But I am very happy to share all the alumni of our department, Biochemistry department especially, to just tell you and share this, that university has honored Professor CV Ramakrishnan at that time with the Lifetime Achievement Award. So it was 26 January 2019 that he has been awarded with this, and I think we should all clap for this, and we should all applaud that this is something like the person who has, if you remember, if I can tell you, from 24 years of his age till 96 years of his age. So such a long duration, giving for the betterment of any particular institute is something. So I think we are all thankful to sir for his all the kindness, all the greatness, and all the vision what he has, and supported us immensely for the development of CMD as well as for the biochemistry department. So with this, I just want to, at the end, I just want to say a happy birthday to Professor CV Ramakrishnan, not only from my side, but from whole CMB family, from whole biochemistry family, all the alumni association members. Very, very happy birthday to you, sir. Many, many happy returns of the day today. And we will be looking forward to have you with us on your 100th birthday. And we would like to celebrate it here with you. And with this, I will thank everyone, and I will invite Professor CV Ramakrishnan to give his talk and discuss with interact with the students and motivate them as every time you have been doing. Thank you, sir.
Excuse me, don't believe anything Professor Sarita Gupta has talked, because she was practically my adopted daughter. So you can understand what a daughter does. So don't believe much of us.
No, this whatever I said is absolutely true. So, sir, we request you to say now, to interact with
Now, I would like to know one thing.
Yeah.
Are the present students watching this meeting or not?
Yeah, they are all there, sir.
Yes, sir, they're all on YouTube, sir.
Excuse me?
They're all there on YouTube.
No. What I'm saying is, if I if I talk,
yeah, they are
hearing. That's what I want to know.
Yes, they are listening.
Because I want to talk more for the students rather than for you people. First of all, I cannot imagine that I will be living at 96 with all my health problems. So it is a wonder. And second thing is, I never thought, for example, excuse me, one more thing. If you know, in most of the institutions, when the fellows were ruling the institution and all, everybody will be saying sir, and all these things. But once when the fellow retires, if he goes to those institutions, as far as I know, even the peon won't get up and say hello to them. That is the way, because the way in which they behaved, like Maharajas when they were ruling, nobody would bother. But what I find now is, actually after my retirement, I find wherever I go, the faculty and the students, they are around me all the times, just make me happy. And that is why I think I must. And now you have celebrated this 96th birthday. Again, I do not know out of the employees of the university, retired employees, how many 96-year-old fellows are still living. I have feelings, maybe I may be wrong. Perhaps I am mostly the one fellow or maybe 1 or 3, 4 fellows living now. So now after so many years, if you call me and then celebrate it, I must be extremely, extremely grateful to you for that. The other thing is, I was very happy when you talked about this meeting because I can have the darshan of all you wonderful people who still look young when I'm getting much older and so. So I enjoy seeing all of you. Now, for the sake of the students, I want to say a few words. One is, you know, this biochemistry department was started with just 20,000 rupees given by Professor K.G. Naik. And when they invited me, I found there was an empty laboratory and they had 4 students admitted. And they told me, you are the only staff, you should teach, take them around to all other departments and then do the practicals. You see, all sorts of things they said. But, you know, due to the famous person of whom you students may not be knowing, Dr. Jivaraj Mehta, who is responsible for starting this university by converting all the colleges into university. He was the finance minister of the Bombay University government, and then he became the chief minister of Gujarat. That man took a great interest the moment he saw me. And he was the man who went all the time to Delhi and saw to it that the University Grants Commission, in the next, within 3 years, they gave us a huge grant to have a large number of faculty, the buildings and everything. That is how this biochemistry department started in a big way. And the interesting thing was, we felt in most of the places, the fellows, the professors will say, I am doing research, and they will completely neglect the teaching. And in many places, they will say, I am teaching, but they will neglect it. In fact, I remember Dr. Jivaraj Mehta told the medical college professors once, friends, at least once in 20 years, you change your notes so that at least something will be fresh in 20 years. That is the way it was going. Whereas regarding our biochemistry department, I tell you, whether you people know or not, there is one Professor C.N.R. Rao, the famous scientist who has developed all the institutes, ISR and everything. And the other man he is no more, Professor G.N. Ramachandran, very famous scientist for his protein structure studies and all. It seems these two people went around and told others, there is only one department in this country, that is the biochemistry department of MSU, which gives equal importance to teaching, training, and research. And that, you know, made me very happy. I have never, of course, I have met Dr. G.N. Ramachandran. I have never met Professor C.N.R. Rao. I know his son-in-law, who is Ganesh, was the ISR, you know, director. But, you know, this is the name the department got. And now, in this department, we concentrated, as I have told you, I you know, lectured till I retired. And you can ask Professor Rao, I was up to date every year because I used to read all the books and their annual reviews, and whatever I talked, It was just the present, not the old rack which used to be. That is what the biochemistry department was known. The other thing biochemistry department was known is, it has, of course, it has become very famous. You see, you know, internationally it has become famous for its research. It got a special center for teaching and research in neuroscience as well as nutritional biochemistry, and it has become really well known. You see, there's no doubt about it. But then the most important thing is, even the PhD students, and they were sent, even while doing PhD, abroad for 1 year to work with the laboratories which were collaborating with it. One of the examples, Professor R.K. Rao, who is here. You see, so like this, you know, these students, they had they had an experience. And then what happened? When they did wonderfully well, afterwards, those fellows invited them to come and work with them. That is how, you see, this department became really famous. Then in 1986, I retired and moved to Madras. I joined the Madras Medical Mission Institute of Cardiovascular Disease as the director, honorary director of academics, and I started a very special program called MS Physician Assistant-MPH, which is only done abroad in United States. This was done in collaboration with BITS Pilani, and even now it is going on wonderfully well. Then in 1996, I left India and then went to stay with my daughter in United States. In 2005, Biochemistry department started the Golden Jubilee, performed the Golden Jubilee, and they invited me. And from that year onwards, I was coming every year to Baroda to move with faculty members and the students at least for a week, and enjoying all the receptions and the entertainments, everything they used to give. Then in 2007, when my wife died, I have given all our money to the Coimbatore Ramakrishna Mission Vidyalaya for giving 5-year college education to very, very poor children of all religions and everything. That means their family income should not exceed 6,000 rupees. And nobody from the family gone to college. So these people, they can study in any field for 5 years, and they were given free fees, boarding and lodging, clothes. They don't have even proper clothes. Then summer training. And now these people, now for that, one of the persons, there are 2 persons here who have donated for it. Tara Mehta, I think she gave some 2 lakhs or so. And our R.K. Rao, $5,000 or $1,000 each. So here, all these friends have given donation and everything. So far, 22 boys and 55 girls have finished college education. They are in good job, and poverty has disappeared from their families. And now, so they are all leading an extremely happy life. Now, from 2010, the Professor Goel invited me to help Professor Sarita Gupta to, run that Center for Molecular Biology. And later on, Professor Yogesh Singh came. And Professor Yogesh Singh and Professor Desai, the dean, they extended extraordinary help, for her to develop the department wonderfully as a unique one. Now, why it is unique? Because Again, the reason is all the departments of science faculty cooperated to teach different courses. That is number one. Number two, all the top research centers in India, as well as the research centers abroad, these scientists have agreed to participate in the program. They came to Baroda whenever possible to give a short course, interact with the students, and take them for training in the summer to their place. I do not think there is any other department like this in India which has got this type of collaboration as well as this type of students going up. As I told you, Dr. Rao showed in his table 2 students from this place, who worked with him. They are extremely lucky. Now Professor Rao says, I am willing to take some more students and take part in the program. So that is how these 2 programs are going on wonderfully well. Now, till 2019, every year I came to India to stay sometime in Baroda, then in Madras, and then mingle with those students in Coimbatore. In 2019, it's not a question of pandemic. In 2019, there was no pandemic. But from 2019 February, I suddenly got unsteadiness, giddiness, and everything. So I have been asked to stop traveling. So even if the pandemic goes away, I don't think I will be able to, travel, but I will always be seeing in the Skype wonderful people like you anytime which you want to interact with me. So I am now living. In fact, actually, I am, once R.K. Narayan said, when his daughter died, I am waiting in the queue online, but my daughter jumped over me. So in the same manner, in the last 1 month, when I was standing in the queue, 3 wonderful people who are known to me, they jumped over and died. So what I'm saying is, I am now extremely happy, pampered by my children, pampered by people like you. And when death comes, I will die happily knowing that all of you are running your programs wonderfully well and the students are very happy. Thank you.
Thank you, sir. Thank you.
Maybe I bored you, but then I'm sorry.
Everybody, please on your camera. Students want to take a group photograph.
What is that?
No, please on the cameras, everybody. The students want to take a photograph.
How can they take the photograph like this?
Yes, sir. Yes, sir.
They can take. No, if they can take, they can take. I am sitting here.
But you keep your head up, sir. Keep your head
What is
up.
that?
Keep your
Head up.
Yeah.
Head is very low. Yeah, that's good. Just up.
Yeah. This is okay. Don't look down. Look up. Yeah.
There you please take the photo.
Yes, ma'am. It's click. Thank you, everyone.
Yeah.
Where is
Is
Dr.
it all right?
Rao? Where did he go? Yeah.
Is it okay?
We will now have the national anthem. If anybody else wants to say something, Jarori, anybody want to say something?
I would like to add about the forthcoming program.
Excuse me?
Sir, we have 3 lecture series in the Department of Biochemistry.
Yes.
CVR lecture series. Professor Telang.
I don't think there is. No. As far as I know, Professor, what happened was once they called it a CVR lecture series, but then when they also put some fund for Telang, I told the CMB or anybody, make it or tell the university, it will be a CVR-Telang lecture series. So I don't know whether there is a separate CVR lecture.
We are holding two different lecture programs.
So I don't know. That's what I'm saying. I know only one lecture series called CVR-Talent lecture series. Again, I have made certain things with certain agreement with the university. The money is with the university. I said only the interest from this fund should be used, annual interest. Again, I said this annual interest should be used for the professors who come with their own money, only to pay for their stay in this, but not for the travel grant. And the university agreed for it if Professor Sardar Gupta is there, and they officially agreed. So that my idea was that fund can go forever. Otherwise, to be very frank with you, in most of the universities in India, each professor will use the money to call his friend from the other thing, and that fund will be over in 2 years. That I don't want to happen for these things. So I don't know about the other thing which you are talking about.
Sir, actually, we have been following what you have said.
Yes.
We held both the lectures in 2018 also. And Professor YJU, Professor Pawan Agarwal, they both gave Professor Telang Memorial Lecture. And for Professor CVR lecture, we invited Mohan Rao from CCMB Hyderabad. He served as director of CCMB. So he was invited. Last year, Professor Daniel Finlay from Harvard Medical School delivered a CVR lecture. And you know about LJ Parekh Memorial Lecture Series. And
yes, that I heard about it, yes.
Yeah. And now this year we are going to hold, on the occasion of National Science Day, a lecture, CVR lecture by Professor Craig Cruz from
Professor.
From Yale.
Professor.
Yes, sir.
No, Professor.
Yeah.
You will be lucky because you need not pay any money. This is all. So that way, no. What I am saying is, if you can get the people to talk without giving them any money, you can go ahead. And again, the other thing is, you know CVR now, but the next person comes after 10, 15 years, he will not know who is CVR. And also, anyhow, we will enjoy just now, you see, and that is it, because that's always the case.
We hope the tradition will be continued and the standards will be maintained.
Continue till this professor is there. No. The point is, now I only want to see things going on wonderfully with you, but I do not want to get involved in anything anymore. Otherwise, I don't want to die feeling I am missing something. I want to die knowing everything is there. I have got nothing else. You see, that way I want to die. Thank you. Thank you very much. It's very nice, sir.
Yeah, Professor K.N. Ganesh, you have mentioned just now.
Yes.
He is going to give Professor Telang Memorial Lecture.
Very good. Very good. That will be fine.
So we have planned this way.
Fine. So are we closing?
We can have the national anthem before we close.
Sure.
As Madam
And Pushpa, thank you very much. You are the one who tried to organize. No. By the way, can I talk some? I'm sorry, I have forgotten to talk something to the students. Can I talk just 2 minutes?
Sure. Yes, sir.
No, okay.
No, sir, you can.
You said no.
No. Go ahead, sir. Go ahead.
You can continue.
Okay. This is mainly for the students who are listening to me. I told you every time when I came to Baroda, our Professor Pushpa Ben, Professor Sarita and all, they showered with me with lots of gifts. Now, for you students, I am asking you to give me a gift since you are celebrating my 96th birthday. Sometime back, I sent to Professor Pushpa, Sarita Gupta, and Professor Ratna Prabha complete details about the Coimbatore project. You see, in that letter, I have mentioned the website about the Coimbatore project, every detail, and requested them to see whether they can print it and give to the student, so that if the students like the project, they can give it to their parents. And if the parents like, they can donate liberally to this Coimbatore project. So I would be very happy if you students manage to take this, and if you like it, give it to your parents. Similarly, I am requesting these three professors to kindly send those letter which I sent also to your old students. My point is, I do not expect everybody to do this, but those who like, they will definitely be helping the poor children. Only when the poverty disappears and every child gets education in India, these disputes between Indians, Muslims, and all will never stop. Thank you very much for giving these 2 minutes. We can go for national anthem.
Yeah, unless somebody wants to say something. Jarori sir, anybody?
What is that?
Jarori sir, you want to say something? Dr. Jarori is here. Yeah.
Oh my God, is it Dr. Jarori?
Yeah.
Is it Dr. Jarori?
Yes, sir.
Yes, sir. Professor, 1 minute. Professor Jarori.
Yeah.
Only a week back, I was thinking of you and wanted to know how your health is, and I am now seeing you. Fantastic.
It's better, much better.
Speak loudly. Yeah.
Huh?
Yeah,
I have to.
close by. I can't raise my voice.
Yeah.
Please sit
Can
down.
you hear me? Can you hear me?
Yes, yes.
Yes, sir.
Yes.
Okay, I just remember an anecdote, so I just want to say that during the TIFR visit by Professor CV Ramakrishnan, he asked me, my father every year wants to come to India. Does that help the department in any way? So my quick answer was that it helps in every way. He drives the program, he drives the syllabus, he drives the appointments of faculty, he goes to Gujarat government, he goes to university office, and he collects the people who are well-wishers of the department. So what else can one ask for? His sheer presence makes the program run. So that's why your coming to Baroda was very, very important, sir. And I hope at the 100th birthday, again, your health will cooperate with you and you will be in Baroda. Thank you.
I am very happy to see you.
Professor Patel.
Yes, I would like to wish you very happy birthday, Professor CVR. There are a couple of things I would like to mention here. That I was in the department from year 1962 to 64, and there are nice to see two contemporary right here on the screen. Dr. Ambrish Patel and Dr. Tara Mehta. I think they were my contemporary at that time in the department. So it's nice to see both of them here. I like to mention two things. That while I was a MSc student, Professor.
Excuse me, sir. Can you please tell your name, sir?
Hello.
Can you please?
MS Patel, sir. Mulchan sir Patel.
MS Patel. Mulchan.
Oh, this is MS. Is it MS Patel?
Yes, yes.
Yes, sir.
Yes.
My God, I couldn't. I'm sorry, sir. I couldn't recognize you.
I'm sorry, my name is not appearing. It just simply says on camera, you. That's something, And
You see, you went and worked with him, right? Isn't it?
Professor Sarita Gupta worked with him, sir.
Yes.
Huh?
Sarita
I
madam
thought,
worked with him, sir.
no, I am sorry. You are Professor M.S. Patel from Buffalo.
Correct.
Oh, yes.
From Buffalo,
Correct.
yeah.
Yes, Professor Sarita Gupta worked with him.
Yes, for 3
Yes,
months
yes.
visit, that she
Yes.
was in my
You
lab.
see, you have become younger, so I couldn't recognize you.
Yes. There are 2 points that I want to make, at this occasion, that Professor expressed great interest while I was in the department as a master degree student. He sent me to Nagpur for a visit to his former students so I can learn few techniques. And the second time was the end of the second year. He sent me to Professor Ganpati to learn some techniques in Ahmedabad. So while I was a student, he was sending me to different labs to learn, various different techniques. That was the one thing. The second thing that sticks to my mind is what he just said, that he always kept himself up to date for teaching purposes. And there is one incident that occurred to me, that while I was taking my second year MSc final exam and doing a practical, there was a viva voice, and there were 3 professors who asked students to come in, and they used to ask questions. And I was asked questions about something that I don't really remember the question, but the answer was that I said something about a paper that I had just read in the most recent JBC issue at that time, that was conversion to mannonyl-CoA to succinyl-CoA. And the way I had read probably a week earlier in the Tara Mehta library, a professor immediately said that is not the case. And I said, I read it because it had just come, a week earlier in the library. And so after my viva voice, he went directly to the library. I had given the issue and everything. He looked at it, he read it, he came back to me. And while I was doing my final examination of practical experiment, he came back, he touched my shoulder and said, M.S. Patel, you are right. I think no professor would do that kind of, not only instant checking, but then giving back to your students, what has been said. So after all these more than 50 years, that's one incident has, occurred that, the professor had not only went back to the library and read himself, but he had come back and, really mentioned to the students while I was still doing my practical examination, And so I think those are the real qualities that you hardly see in academic life. And that has really touched my, whole enthusiasm for, being up to date as much as possible, and then recognizing student contribution. So thank you very much for, what I...
I just want to add something to what Professor Patel said. I still remember that Viva exam. And for your information, the person who was one of the examiners was Professor Pai from Medical College. He is no more alive. Whether it is M.S. Patel or others, I don't know. When he asked the question, exactly the student said, "You have entirely new things studied in the journal." That fellow said, "That's not the answer." I told Dr. Pai, "I am sorry. The student has read from the journal and it is correct and you are wrong." This I remember. I don't know whether this or with something else, but exactly I remember this incident. You see, yes, that is the one piece. Now, again, I want to, since you allow, maybe I can talk 2 minutes more. You must know one Professor Acharya who is working in the biochemistry department. That Professor Acharya has recently written to me, "Sir, I used to think that the students, when you were there, they were not, feeling very happy with you and all. But sir, I want to tell you now, all the students who are coming now, these old students, etc., they say that now they know that you are the kindest person. You are strict, but you are very kind and you cared for the students. That made me very happy. Thank you. Anyhow, I think we'll stop because we'll continue.
I think we'll stop with the national anthem. Thank
Yes,
you
yes,
so
that's what I
much.
say.
So this is a good idea. We can have a virtual alumni meet. This is definitely a good idea. We'll have the executive meet and then we'll have the alumni meet. Thank you so much, everybody. We'll stand up for the national anthem. Dhairya, please play the national anthem.
विनायक जय हे भारत भाग्य विधाता पंजाब सिंध गुजरात मराठा द्राविड उत्कल बंगा विंध्य हिमाचल यमुना गंगा उच्चल जलधि तरंगा तव शुभ नामे जागे तव शुभ आशीष मांगे गाहे तव जय गाथा जन गण मंगल दायक जय हे भारत भाग्य विधाता जय हे जय हे जय हे जय 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 हे
Thank you, everybody. A big thank you to the students who did everything, especially Dhairya and the students who made the video for Professor CV Ramakrishnan. Thank you so much. Have a great 2022 ahead.
Yeah, it was really nice meeting and seeing everybody, you know.
Yes.
So,
Nice seeing you all.
yeah.
Take care and stay safe.
Bye.
Also, you gave a credit to Tara too much. It is a Hansa Mehta library, not Tara Mehta library.
Sorry about that. Yes.
I
Hansa
remember
Mehta
all of
library.
it. Yeah. Sure.
Thanks for correction. I'm getting old now.
We are not.
Okay, thank you, everybody.
Bye.
Bye.
We'll have general body meeting sometime.
Yes, madam. Yeah, okay.
Yeah.
Thank you all.
Thank
Thank
you.
you, everyone.